So, hello everyone. Very good evening to all of you. So, how are you all doing, and how is this Brahmastra series is helping you all out? I'm pretty sure that this series, like the kind of questions that are being discussed in this series, is definitely helping you out in solving the questions. Not only solving the questions, but also you are learning how to approach a particular question. Am I correct? Good evening, beautiful colors. So, in today's session, I have taken two topics. That is, one is cytoskeletal system and another one is genome organization. So, in the recent past, if you see, cytoskeletal system plays a very important role overall in Unit 2. Earlier, this particular topic was ignored, being a part of 2B, which is basically consisting of cell organelles. Major focus was on other cell organelles, but this part, that is the cytoskeletal system, was largely ignored. But recent past, we are getting good number of questions, and that too those questions are in the form of 4 marks. So without wasting much time, let's go to the first question from cytoskeletal system. And let's take up a very simple question which was asked recently. Cells are physically linked to one another and to the extracellular matrix through their cytoskeleton, and this imparts strength and rigidity of the tissues as well as organs. Most of the animal cells have three types of cytoskeletal filaments. I guess you all are aware, and that is also mentioned in column A. Their corresponding functions are listed in column B. So, from the given options, you have to identify the correct combination from here. Find out. And most of the times, if you see, questions are coming in the form of match the following only. Special focus on accessory proteins. So I'll come to that part also. Just started with a simple question so that you'll get that confidence to move ahead. Yes. So we have intermediate filaments, microtubules, actin filaments, the three major cytoskeletal proteins in our system. Determines the shape of a cell surface necessary for locomotion, maintains the position of membrane enclosed organelles, providing intracellular transport, and the third one is providing mechanical strength of a cell. Okay, I can see beautiful colors has given the answer. So can I expect some more answers from others as well? And let's make this more interactive. Let's make yourself more confident in giving the answers. Yes. Can I expect more answers? I can see many participants are here, but getting only one answer so far. Try to participate. As I always say to everyone, this is not a final call. You are attempting out something. You are trying to identify your strength and weakness. So if you're giving a correct answer, obviously you get that confidence that, yes, I know this part. You can confidently say. But if you're giving the incorrect answers, then also no problem. You can learn from the mistake so that you will not make the same mistake again. Right? Okay, yes, Bharata, I can see your answer coming up. Some more answers. Come on, everyone, participate. Let's check yourself out. So, this is a kind of checking out your own strength and weakness. Yes, give me the answer for this question. And this is a very simple question. I don't think so. This is that much tough. Basic, the three proteins that comprises of cytoskeletal filament. Yes. Okay. So let's take up the answer now. The correct answer over here is, as you both have mentioned, absolutely correct. Option 3. So what does this option 3 stands for? So always remember, intermediate filaments is something that is not involved in movement. Of course, there can be a confusion that microtubules are involved in movement, actin filaments are also involved in movement. But intermediate filaments does not have that role. It is just providing us with the mechanical strength of a cell. So you can easily opt for this option. You can easily eliminate the first and second option. Now there will be a tug of war between 3 and 4. Next, you need to identify how to filter out between microtubules and actin filaments. Always remember, another unique feature of microtubule is that they are the one which is responsible for positioning the membrane enclosed organelles. No doubt, whether you take into consideration microtubules or actin filaments, both are involved in movement. But the major function of microtubules is positioning the corresponding cell organelles at a desired location. And the third one, that is the actin filaments, it determines the shape of a cell surface, thereby necessary to carry out the movement. Okay, so the key word over here is positioning of membrane enclosed organelles. Now, as I told you, we have got three major cytoskeletal proteins. So, microfilaments, they are made up of actin monomers. Microtubules, they are made up of alpha beta tubulin dimer. And intermediate filaments, depending upon the location, different proteins are involved over here. So, can anyone tell me how these proteins are interacting with each other? What is the bonding between them? Like if I say microfilament to microfilament, microtubules to microtubules, or intermediate to intermediate, what is the bond they share? Or what kind of bonding they will be having? Can anyone say? Like we have hydrogen bonding, hydrophobic interaction, covalent interaction, non-covalent interactions. So what kind of interaction do you find in this cytoskeletal protein? Anyone? Any idea? No? Always remember, all these filaments will undergo frequent assembly and disassembly. Right? When required, they will be assembled, and when required, they will be disassembled. So, each of this cytoskeletal protein is a polymer of protein subunits which are held together by weak non-covalent bond. So, why it is a weak non-covalent bond? Because then only it will be easier for them to undergo quick assembly and reassembly. Right? So, that is a part of our cellular regulation. Absolutely correct. Beautiful colors. So this is a comparison chart we can always remember whenever you go for the exam. So we have microtubules, intermediate filaments, actin filaments. So when I say subunits that are incorporated into the polymer, alpha beta tubulin, which is a kind of heterodimer. Here, as I told you, different proteins will be there. And in this case, it will be ATP actin monomers. Preferential site of incorporation, that means where the corresponding protein has to be incorporated. So when it comes to microtubules on actin filaments, in both the cases, obviously it will be the plus end. But remember, in case of microtubules, among the alpha beta subunits, beta tubulin is the part where the addition takes place. And when it comes to actin filaments, the positive end is known as barbed end. Any guesses what does minus end indicates here? What does minus end indicates here? The way positive end is known as barbed end, for minus end, what is the name over here? Here we can say that, okay, alpha tubulin is the minus end. That is quite obvious. But here what we can say? This is known as pointed ends. Minus end is known as pointed ends. Remember this part. Polarity, yes, they do have polarity. Which one? Both microtubules and microfilaments. Enzymatic activity, yes, GTPase is there. Here ATPase is there. What are the motor proteins involved? Motor proteins means that help in the movement. When it comes to microtubules, we have kinesin and dynein. Intermediate filaments, as I already told you, there is no locomotion over here, so there is no such proteins. For actin, we have got myosin protein. Then various associated proteins that I will discuss in details in the upcoming slides. This is their structure, and dimensions is not important. Distribution, yes, they are present in all the eukaryotes, both actin and microtubules. But intermediate filaments, they are present exclusively on animals. Okay. So this is the basic comparison between all the three major cytoskeletal system. Remember it. Next, moving on to our next question. Measurement of the rate of actin treadmilling in vivo shows that it can be several times higher than it can be achieved with pure actin in vitro. The treadmilling in vitro can be enhanced by providing profilin that binds to the G-actin on the site opposite to that of the nucleotide binding site, coffilin which binds specifically to the ADP containing F-actin, destabilizing the filament, buffer with ATP with low levels of cations, or buffer with ADP with low levels of cations. Identify the correct combination of statement from here. Identify. This you can answer if you are confident with profilin, coffilin, the role of cations, ATP and ADP. So if you're clear with this terminologies relevant to the system, I mean the cytoskeletal system, you can get into an answer. Yes. What is the function of profilin in general? Or what is the function of coffilin in general? Think. Yes? Can I get some answer for this question? Okay, I got an answer from beautiful colors. Can I get some more answers? Yes. Everyone. Can I get some more answers? Let's there be more of interaction between us. Yes, can I get some answers? So I hope so. You know what is the difference between G actin and F actin? Anyone? What is the difference between G actin and F actin? G actin is basically the monomeric version, right? Actin filaments are present in the monomeric form. But when your monomers combine together in the form of trimers, the trimers keep on adding on themselves. Then you get this F actin. We call it as a filamentous actin. So that is the difference between them. Now looking on to the cations, does cations play any role in this actin treadmilling? So what is the treadmilling? Treadmilling means it's a continuous process. That means continuous formation and continuous hydrolysis takes place. Yeah, exactly. F actin will be considered as a filamentous actin, so that will be a long chain. Okay, so it's like monomers keep on adding on themselves, becoming a trimers over here. That is the thing. Okay, so the correct answer over here should be, as you mentioned it correctly, that is A and B. So now let's check it out. What is the function of this profiline and coffilin? Profiline helps in the exchange of ADP for ATP. Now, what does that mean? Have you heard about this term anytime before? GEF, Jeff. Have you heard about this term anytime before? Maybe somewhere in the unit 2 protein targeting, nuclear transport. Anywhere you have heard about it? Jeff. It is nothing but where exchange takes place. Now, the relevance of why I mentioned the Jeff over here, because profiling somewhere it acts like a Jeff, because it exchanges the ADP for ATP so that it can enhance the polymerization process. So, faster treadmilling can be carried out. Coffee they go and bind to the ADP containing F actin. So we all know that. Yes, absolutely correct. RAN protein, yes, we have that. So coffee when I say coffee they go and bind to the ADP containing F actin. So ADP means obviously you can identify that this is the inactive version or the breakdown version. So when it is connected to actin, so obviously you can guess they are destabilizing the actin filament. Okay, so this will happen towards the minus end, which induces the severing and depolymerization. Next, buffer with ATP is okay, but they require higher levels of cations. So basically, they are cation dependent process. Okay, so actin polymerization requires physiological levels of cations. Low levels will reduce the filament formation as well as treadmilling. So if C is incorrect, obviously we can expect D is also incorrect. Right. Now, if you look into this image, what is this? This is your actin filaments. So what is coffilin? So we call it as a coffilin cycle, profilin cycle, and a thymosin cycle. Now what does this 3 cycle indicates? Cycle of coffilin, as I told you, they will be binding towards the minus end of the actin filament. So minus end means obviously involved in destabilization. So this increases the turnovers of actin monomers by enhancing the disassembly at the pointed end. So when you are degrading these filaments, you are getting the monomers. So you are saving the monomers for the future treadmilling. When it comes to profiling, as you can see, it is a ATP-ADP exchange. So due to this exchange, it contributes towards the barbed end. So polymerization and filament elongation takes place. And the third one is your thymosin protein. So what does this thymosin will do? They will sequester your G protein or the G actin. Sequester means they hold the G actin so that it can use it for future use. I mean, when they require, they can be utilized. So they are preventing the polymerization event, which helps in maintaining a pool of actin monomers for rapid use whenever the cell requires it. Okay, so that becomes your cycle of, thymosin cycle. Okay. Moving on to our third question, an investigator expresses a GFP fused protein that localizes to the outer membrane of Golgi apparatus. Upon visualizing GFP signal in the fluorescence microscope, it was noted that GFP is pericentrosomal in its location. So, treatment of such GFP expressing cells with a newly identified drug disrupted the Golgi into small vesicles. So, here a list of potential targets is given. You have to identify the correct combination of the target. So, figure A indicates where the GFP is located in the pericentrosomal location. Figure B indicates what? They are being provided with a drug because of which they got disassembled or disrupted. Now you need to identify what will be the target of the drug. Dynein complex, myosin, microtubules, or a dicer. Single answer or a combination of options. Identify. So sometimes you can also expect experimental questions from this. But again, experimental questions, very less they are asking most of the times. I mean, in the recent past, if you look into the question paper, direct questions are only they are asking, specifically focusing on the accessory proteins. Yes, looking into the image, looking into the potential targets, identify if you are able to correlate or not. Yes. Now this is also a very simple question. Many a times it happens that whenever we look into an image or a graphical representation, we often get scared and think that this is a tough question, so I should leave this. So never do that. Whenever an image based question comes or a graph based question comes, remember you can sit patiently and have a rethink so that you can get into a conclusion. Sometimes the answer is there, will be there in the question itself. Okay, so don't just leave it off. Yes, apart from beautiful colors, I expect some more answers. I can see a lot of participants are there, but response is very less. So I hope so you understood the first two questions very well. Of course, you have given the right answer, but the explanation part, you have understood it well. Everyone. Yes, Bharata, can I get some answer from your side? I saw you have given the first answer. So if you can try on with this question with some answer. Just whenever you come across this question, these kind of questions, try to read it a few times. Sometimes what happens is that when you keep on reading the question for 2 to 3 times, an answer clicks to your mind. So that is one way of getting into a right answer. Yes. Any other answers? Participants, can I get some more answers? So what you said is absolutely correct. Beautiful colors. So So A and C are the answer. See, this question is so simple. If you are aware of what is what is the function of a microtubule is, they are positioning the positioning the cell organelles over here. So when I say microtubules over here, the two relevant things will be dynein and microtubules. Any idea what is the basic difference between kinesin and dynein? Any idea what is the basic difference between kinesin and dynein? Like which ended motor protein are they? Positive, negative? Which side are they? Again, you might be aware that different cell organelles are located in different positions within the cell. So, some are present in the positive side, some are present in the negative side. So, how this is maintained? This is maintained because of these two motor proteins, kinesin and dynein. So, kinesin and dynein, what is their mode of action? Can anyone say that? Which ended motor protein is kinesin? Which ended motor protein is dynein? Yes? No? Kinesin will be positive end directed motor protein. That means they carry the contents towards the positive end of the cell. Dynein will be negative end directed motor protein. I think the other way around, beautiful colors you have mentioned. Kinesin, remember, always they are positive, right? C. Dynein will be taking the microtubules towards the minus n. So dynein will be negative, kinesin will be positive. Remember this part, okay? So we can easily choose dynein and microtubules from here because myosin is related to your actin filaments. So you can ignore this. And dicer has got no relevance to this question itself because dicer is what? It is related to your RNA interference part. So it has got no relevancy towards this question. You can ignore that part. So like this, you can get into a conclusion. So dynein is a motor protein involved in the retrograde transport along the microtubules, moving the cargo towards the minus end, that is towards the centrosome. And microtubules, these are obviously involved in maintaining the Golgi structure and positioning within the cell that I have already mentioned with the first question. Moving on to our next question. In an attempt to study the transport of secretory vesicles containing insulin along microtubules in cultured pancreatic cells, how would the treatment with colcimide is going to affect the transport of these vesicles? So somewhere this question is relevant to the inhibitors, right? So they have mentioned microtubules. How the treatment of colcimide can affect the transport of vesicles. Which vesicles? Secretory vesicles. So secretory vesicles usually comes from where? Usually comes from the Golgi complex only. So what should be the answer over here? Colcimide induces polymerization of microtubules, which in turn would activate the vesicular transport. Polymerization of microtubules is inhibited by colcimide, which in turn inhibited the transport of secretory vesicles. Colcimide inhibits the vesicular trafficking through inactivation of V snare protein, or they will be activating the T snare protein and in turn activates the vesicular transport. What should be the correct answer to this question? So, in simple words, what they're trying to know is what is the mode of action of colcimide here? Answer I have by mistakenly told. So, I don't know how many of you have heard it. attentively. If you have heard it attentively, definitely you can get into the answer. Mode of action of colcimete on microtubules. Yes, what should be the answer over here? Okay. So beautiful colors has given the answer. What about others? Any other answers to be expected for this question? So this is literally not so tough question unless and until you are aware what is the mode of action of colcimate is in general, and what does this colcimate do to microtubules. So if you are clear with that, obviously you can get into an answer. So here we go. The correct answer over here is statement 2. As I told initially, if you have heard it properly, I told you that you have to remember the inhibitors of microtubules to get into the answer. So, colcimide is basically the inhibitor of microtubules, which in turn inhibits the transport of secretory vesicles. See, it is a drug that inhibits the microtubule polymerization. How? By binding to the tubulin subunits, that is your alpha beta subunits. So, this in turn prevents their assembly into the microtubules. Since microtubules serve as a track for intracellular transport, their depolymerization with the help of colcimate can disrupt the movement of secretory vesicles, which contains the insulin within it. So this disruption hinders the ability of the vesicle to reach their target destination for insulin secretion. So it is considered to be an inhibitor over here. I hope so you got it. Why option 2 is the correct answer. So the drugs that affect the actin filaments and microtubules, if you take into consideration, we have some actin specific drugs here and we have some microtubule specific drugs over here. So actin specific drugs include phalloidine, cytochalasin, svinholide, lantriculin. So what is their mode of action? Phalloidine, it goes and binds and stabilizes the filament. Cytochalasin, it caps the filament plus end. Svinholide severs the filament and lantriculin binds to their subunits, thereby preventing their polymerization. So these are actin specific drugs. Look into the microtubule specific drugs. Taxol, colchicine and colcimate both have the same action. Vinblastine and vincristine also the same action. Nocodazole also the same action. So all the 5 drugs will go and bind to the microtubule subunits, thereby preventing their polymerization. So basically they are involved in disassembly, excepting your Taxol that helps in the stabilization of microtubules, and this is also an example of an anti-cancer drug. Okay. Then actin has got different functions that you have to remember it. So this I have taken from the book itself. So here it is mentioned depolymerizes, stabilizes. So it is clearly mentioned it over here. You can also check on to this table. So whichever is convenient, you can have a look on to that. Okay. So this is just a reminder that we have the Lakshya batch going on, and whoever has enrolled with Lakshya batch, who will be enrolling with Lakshya batch, will be getting free molecular biology internship program, the classes. And you know that nowadays molecular biology is so important in every aspect of research. So apart from that, you will get the unlimited classes, 24/7 chat support system, your study portal where all the recorded lectures will be there. Test series will be conducted, that is weekly as well as monthly, in order to check yourself out. Conceptica series, that means Some smart tools will be available that will help you to keep yourself in track and save more time at the time of revision. So, anything more you want to know, you can always call us in this toll free number displayed over here, or you can visit to our stores.biotechnica.org. Okay. Next, moving on to our next question. Now, this is related to actin binding proteins regulating the microfilament turnover in a eukaryotic system. Match the actin binding proteins with their function. So they have provided with you some accessory proteins in column 1 and their function in column 2. You have to identify the correct combination here. You can take this as option 1, option 2, option 3, and this as option 4. So this is a recently asked question. Let me see how many of you can give the answer. Recently asked question. As I told you, accessory proteins are very important when it comes to examination point of view. That is, whether it is your microtubules or it could be your microfilaments in both the cases. So, what should be the correct answer over here? Any idea? Yes, any idea regarding this correct combinations related to actin binding proteins? If you don't know, I will directly show the answer. We'll display some more examples of this actin binding proteins. Are you trying it out, or shall I display the answer and give you the explanation? Okay, beautiful colors. Okay, so you have given the answer. Any other answers from anyone else? So the correct answer over here is very good analysis. Option 3 is the correct answer. So for this, you need to be clear with each functions of accessory proteins. So profiling, I have already told you that they are involved with the exchange. So you can easily opt for B for 1. So if you are lucky, then among the given options, you have only this B1, B to 1 as one such option, then you can easily fetch a 4 marks, right? Other than that, you can check for other things like coffeeline, which is responsible for the breakdown, right? So that will be binds preferentially to filament that contains your ADP actin, which I have told you just now. Thymosin, they are responsible for G actin binding and inhibit their addition. And lastly, we have got formin, which is responsible for assembly of unbranched filaments. So let's have a look into some examples. So providing you with the examples of accessory proteins involved with microtubules first, then I will go for microfilament. So gamma Turk tubulin ring complex, they help in the nucleation, that is assembly, and remains associated with their minus n. Try to remember what is the name of the protein and in which direction are they acting on. Statmin, it prevents the assembly. They will go and bind to their alpha beta subunits, prevents the assembly. But this is quite opposite to of plus tip. What does the plus tip will do? This will remain associated with the growing end, that is the plus end, and can link it to some structures, for example, membrane. Then you have got kinesin 13. So remember, kinesin 13 and XMAP 215, they will be acting on same side of the microtubules but with different functions. What is that? Kinesin 13 involved in the disassembly, or we can also call it as a catastrophic event. Whereas XMAP 215, they stabilizes the plus end. So one is removing, one is adding. So accelerating the assembly. Catenine, they break down the microtubule. MAPS, microtubule associated proteins. As the name indicates, they will be stabilizing the overall microtubules. Then you'll be having some filament bundling proteins, some cross-linking proteins, some can attach with other cytoskeletal system, etc. So these are examples of microtubules. Now, if you look into the accessory proteins related to microfilaments, as I told you, we have formin. So they will go and bind towards the plus end, that is the growing end, and they help in the nucleation. Nucleation means assembly only. ARP2 bar 3, they are involved in nucleation, but remember they will form web-like structures, or you can say branched structures. These are involved in unbranched structures. These are involved in branch structures. Thymosin, they will go and bind to the subunits, preventing the assembly. Profiling, as I told you, they help in the elongation. Tropomodulin. So, tropomodulin, tropomyosin, not to be confused. You can remember tropomodulin means some modulation happening over here, where they are acting towards the minus end. Tropomyosin, they are stabilizing the overall filament. Gelsolin is simply like your catenine that breaks down the filaments. Coffulin, they bound to the ADP actin, accelerating the disassembly. Then, of course, you will have some capping proteins present either in one end or sometimes present in both the ends, preventing their assembly and disassembly. So, here also you will come across some filament bundling proteins, cross-linking proteins, and some proteins which are attached to the membrane like this. Okay. So, these are the accessory proteins. So, now you got to know that different types of accessory proteins are there. Now, based on this, check out if you can answer this question or not. Cellular actin levels can be as high as 100 to 400 micromolar, and of this, unpolymerized actin concentration can be as much as 50 to 200 micromolar. However, the critical concentration of actin polymerization under in vitro is about 0.2 micromolar, and some of the proteins inhibit the polymerization of actin. Now, you have to identify the list of inhibitors out of this. Thymosin, capping protein, tropomodulin, XMAP 215. Identify the inhibitors out of the given options. Based on your understanding from the previous slides wherein I had discussed about different types of accessory proteins, can you identify which of them are the inhibitors? Remember, they are asking you the inhibitors for actin, that is microfilaments. And in this question, inhibitors indicates the function of disassembly, the breakdown of actin filaments. What should be the answer over here? List of all the inhibitors. Absolutely correct. Beautiful colors. Because you can, from this, you can easily make out XMAP 215 is a activator or a stabilizer related to microtubules. So with this one word, you can eliminate this one. And rest of the things, they are related to actin. Modulin, just now I said modulation, capping, inhibition, thymosin, inhibition. So all are these inhibitors of actin filaments. So you can easily opt for option 1. So that much easy is this question. They have just mentioned these numbers just to confuse you, because we have a tendency that, like many of us have a tendency that whenever we see the numericals, we get bit tensed. So what is this question? But this numericals are nothing. You have to simply identify the odd man out. That's it. So this is just the explanation. Anyways, you already know. Now let's take up the next part which I was mentioning, that is genome organization. This is a recent question from Genome Organization. Following statements are made regarding the nature of chromosomes in bacteria. Most of the bacterial chromosomes are circular. In few bacteria, even linear chromosomes do exist. All the bacterium systems are known to have a single chromosome. Some bacterial chromosomes contain enhancer-like elements. Chromosomes in bacteria are stabilized by histone-like proteins. You have to identify the correct combination of statements from here. Identify what is the correct combination of statements over here. Very simple, very direct question, and I remember this has been asked in the last exam, 4 marks question exam. Part C. Of course, we are focusing more on part C questions only, but today I have also kept some part B just to make your concepts more clear, specifically related to genome organization. That's it. So this is a 4 marks question. Okay. So beautiful colors has given the answer. The answer over here is 2. The answer over here is, it's not ACD, it's not BCD, rather it is ACD. All the bacterial systems not necessarily should have a single chromosome. Just check it out, Vibrio cholera, they have got 2 chromosomes. So this is not mandatory, but rest all are correct. Most bacteria will have a haploid genome, a single chromosome consisting of a circular double-stranded DNA molecule. However, linear chromosomes have been found in gram-positive bacteria like Borrelia as well as Streptomyces species. One linear and one circular chromosome is found in gram-negative bacterium, that is your agrobacterium. Bacterial chromosomes not having exactly the histone proteins, but they do have histone-like proteins in order to stabilize the chromosomes. And that's the reason correct option over here is A, C, and D. Just have a minor clarity of this question, fetch a 4 marks from here. This is a 2 marks question, but yes, I want you to answer this because it requires some conceptual clarity which will be helping you out in solving 4 marks. Which of the following statements about chromatin is not true? DNA winds up approximately 1.65 times around the nucleosomes. H2A, H2B bind both the entry and exit ends of the DNA in the nucleosomes. Covalent modification of the histones influence the chromatin compaction. Non-histone proteins are part of mitotic chromosomes. Remember, here you need to identify the incorrect statement. So whenever you get a question, read the question thoroughly to know whether they are asking the correct statement or the incorrect statement. Sometimes out of excitement, we jump into the answer and the question was not true. We get into the answer which is true. So that is a very common mistake we all make. So that should be avoided simply by go through the questions thoroughly. So what should be the answer over here? Now this is a 2 marks. Everyone should answer this question. So whenever, it's a request to all of you, is that whenever you are attending these QA sessions and all, try to be more alert, try to be attentive, and most importantly, try to be more interactive. As I am repeatedly saying in my previous session also and in this session also, this is for you. These sessions are done for you only. to make you realize where are you going wrong, or to give you that confidence that you are doing correct. So it's very important that if you are attending the sessions, try to interactive more. Okay, you have opted for option 2. Is there any other answers? Anyone else? Anyone else? Let's take up the answer. The correct answer over here, as you mentioned, absolutely correct, option 2. Now let's check it out why option 2 is incorrect. Nucleosomes from all the eukaryotes contains 146 base pairs of DNA wrapped. That is true. But the thing is that the linker DNA can be variable among the species, which could be a range of 15 to 55 bases. So that's the reason when you're combining this histone octamer with that of an H1 protein, your total wrapping of DNA changes to 168 base pair approximately. That is depending upon this size. Linker histones, that is H1, bind to the nucleosome at the entry and exit side. So this clarifies the fact that it is like present in the entry and exit side. H2A, H2B is not present. Rather, what is present? Your H1 is present. And it allows the formation of higher order structure. Okay. Now, this is different levels of organization of chromosomes. So, at the simplest level, remember chromatin is a double-stranded helical structure of DNA. DNA, when complexed with histones, we call it as a nucleosomes. So, each nucleosome will be having 8 histone proteins wrapping around the DNA 1.65 times. Okay, so as I told you, based upon your linker DNA. Then we have got a chromatosome, which consists of a nucleosome plus the H1 histone. So when they combine together, we call the structure as a chromatosome. So this nucleosome will undergo further folding to give rise to 30 nanometer fiber, which then form loops, that is 300 nanometer in length, and they will be further compressed, giving rise to 250 nanometer wider fiber. And finally, you see the metaphase stage chromosome, which is the highly compressed stage, that is tight coiling of 250 nanometer, giving rise to a chromatid of a chromosome. You all might be aware that different stages, different names are given. So if I say first order of packaging, we call it as a beads on a string-like appearance. Second order of packaging, that is the 30 nanometer fiber solenoid. When it forms the loop, we call it as a scaffold loop. Then condensed form is the chromatid. And finally, we get the chromosome. That is the metaphase stage. Another simple question to you, again a 2 marks question, but yes, it holds a good significance for the basics of genome organization. Which of the following statement is not true about nucleosomal organization of core particle? The typical structure of the DNA is altered in the middle of the core particle. DNA is organized as flat superhelix with 1.65 turns around the octamer. While forming 30 nanometer fiber, 6 nucleosomes per turn organize into a 2-start helix. The N-terminal histone tail in a core particle are strictly ordered and exit from the nucleosome between the turns of the DNA. Again, identify the incorrect answer from here based upon the nucleosomal organization. Yes? These 2 marks questions forms a base to construct a 4 mark answer. Because every time you get a 4 marks question for sure from genome organization. And they are asking very basic questions. So you have to be very clear with every nook and corner of how this nucleosomal organization takes place. And that's the reason, even though we are focused on part C, but specifically for this topic, I have taken two part B questions just to ensure that you should get a conceptual clarity on this topic. What should be the answer for this question? Anyone? Any idea? Guess? No? No answers? No one is able to attend this question? Let's take up the answer. Otherwise, you will not get to see further questions. We are just left with 6 minutes now. So the correct answer over here is 4. So 4 is incorrect. The N-terminal tails in a core particle are strictly ordered and exert from the nucleosome between the turns of the DNA. Now that is incorrect. See, N-terminal histone tails are flexible and unstructured, so they will be extending outward from the nucleosome core. And also, it is in the N-terminal region you will find that most of the post-translational modifications takes place. That is, epigenetic modifications takes place. Then chromatin structure regulation, that is whether to switch on the gene or switch off the gene, that is also carried out based upon the kind of modification. I guess you all might be aware that we have acetylation, phosphorylation, those things are going on. So where does it happen? Towards the N terminal of the protein. So while assembling a nucleosome, remember the histone folds first binds to H3, H4 and H2A, H2B dimers, and H3, H4 dimers combine together forming a tetramer. Now this tetramer will be combining with two other dimers. They will form a compact octameric structure around which the DNA is bound. Now we have how the bonding takes place. Hydrogen bonding will be happening, that is 142 around between the DNA and the histone core. Now how this hydrogen bonding takes place? Between the amino acid backbone of the histone to that of the sugar phosphate backbone of the DNA. So that is how the interaction goes. Apart from hydrogen bonding, hydrophobic and salt linkages also contributes to the histone protein and DNA together within the nucleosome. So this is how it goes. We have a tetramer, we have got a dimer. Now slowly the DNA comes and binds to it. So when the DNA is bound, all the histones will come together and you can see the N-terminal tails are exposed outside through the DNA. Why? Because as I told you, the major reason is they need to undergo the post-translational modifications over there. Okay, I hope so I'm clear with this question. Moving on to the last question. The nucleosome is the fundamental subunit of chromatin in eukaryotes. So following are some statements provided. A typical nucleosome contains 200 base pairs of DNA, 2 copies of each histone, 146 base pair of DNA per nucleosome core particle. It is strictly maintained across the organism. Octamers are not conserved during or after replication, but tetramers are. Variants are being identified for all the core histones excepting the histone H3. While wrapping around the core histones, structure of DNA is altered at the middle of the nucleosome core particle and exhibits increased number of base pairs per turn. Now you have to identify the correct combination of statements from here. Identify. So these are the kind of questions you can expect from this topic in the exam. So if you filter out, there will be more such questions, but since it's already going to time out, like it's going to be 7 p.m., so I cannot take further questions. So try to get an answer for this question. Can I get more answers for this question? Can I get everyone's answer for this last question? So this is basically a summary of whatever I have discussed with you so far. Based upon that only, the question has been done. So now you are able to understand why I have discussed those two questions with you. Yes, what happened? Beautiful colors. I did not get your answer. Previous question and this question also. And others, what should be the answer for this question? Yes, no answer. The last question, not able to connect. Answers I have already told you. Okay, good to see your answer back. Beautiful colors. But the correct answer over here is ACE. That is, a typical nucleosome will contain 200 base pairs. That's what, depending upon the corresponding, what I can say, the linker DNA, this base pairs will be changing. Then histone octamers are not confirmed, but tetramers do. They are conserved. Variants are identified for all the histone proteins excepting H3. They said no. For H3 also we have got variants available. So the structure of DNA, obviously it will be altered in the middle of the nucleosome particle and there will be increase in the number of base pair, which is the correct answer over here according to the previous question if you have seen it. So the length of the DNA, so statement B doesn't require any explanation because as I told you, based upon the linker DNA, the number base pairs can vary. And also if the histone variants are present, for example, H2AZ, that is the variant of H2A. So there also you will find variations in the length of DNA being wrapped. So while the length of the DNA per nucleosome is relatively conserved, there can be variations depending upon the genomic and epigenetic context. Several variants are visible for all the histones. So for H3, you can see H3.1, 3.2, 3.3, SNPA, 3.x. So many examples of variants are seen. So with this, we are winding up the session over here. So that's thank you so much, beautiful colors, for that good interaction and trying to attempt the answers for all the questions. So keep on studying like this, keep on interacting like this in all the sessions. And this is a request to every one of you, to each one of you, to interact, to give answers, and so that you will get that confident on how to work out the question. Thank you so much, everyone. See you again in the next session.